So hello everyone, I hope you all are enjoying the online sessions which are directly coming to your devices. So stay home, stay safe and keep learning. In campus we have started the topic that is absorption. In online session we will continue the topic related to absorption, but prior we will have a quick recap and revision of previously discussed topic, right? So our today's topic is mechanism of drug transport. Right, so let me first introduce you all with the subject that is biopharmaceutics and pharmacokinetics. In biopharmaceutics, what we do, we have a concept prior to the subject. The concept is that there are various sites of administration. From these sites of administration, the drug is moved to systemic circulation. Now, if the quantity of drug, the amount of drug is same and is given from various site of administration, the drug response is not same. Why? Because the rate of absorption, that is the movement of drug from site of administration to systemic circulation, is not same when given from various routes. Right? For example, in case of parenteral, the drug is directly transported to systemic circulation without any barrier. So the absorption rate is 100% and the response is much more faster. But in case of oral, the drug need to be transported through barrier of gastrointestinal tract, that is GI lumen. So the absorption in case of oral is dependent on several factors. There are various factors which influence the rate and amount of drug transport. So when we study these factors which influence the rate and amount of drug transport, and we utilize these study in order to optimize the therapeutic efficacy of the drug product. Right? So this phenomena or we can say this kind of study is known as biopharmaceutics, where we study the factors which influence the drug product, which influence the absorption rate or the rate of drug transport, and we utilize these factors to optimize the therapeutic efficacy of the drug product. Okay, and pharmacokinetic, it is the kinetics, or when we study the rate of ADME, that is absorption, distribution, metabolism and excretion, this study is known as pharmacokinetics. Alright, so moving ahead towards the topic of today's session, that was mechanism of drug transport. In mechanism of drug transport, I can mention mechanism of absorption as well, but rather than absorption I have mentioned drug transport. Why? Because the drug is not directly absorbed when it is given through GIT. It have to be passed through a barrier made up of GI epithelium cells, which is made up of phospholipids. So it is known as drug transport. So the drug is transported via three different processes through the GI epithelium that you can see on your screen via schematic diagram. The first process is transcellular mechanism. The transcellular mechanism itself known as across the membrane. When the transportation process of drug take place across the membrane or within the membrane, it is transcellular. Second one is paracellular. In paracellular, the drug is transported between the linkage of the two membrane or tight junction between the two membrane, right? And the third one is vesicular. In case of vesicular, the drug is transported through the vesicles via process known as endocytosis. So on your screen, you all can see the broader classification of mechanism of drug transport. The transcellular is again divided into two parts, that is active transport mechanism and passive transport mechanism. Similarly, paracellular and similarly vesiculars are again classified into two different processes. So in the upcoming lectures, we will discuss all these processes one on one. And today we will start with our topic with passive transport mechanism and active transport mechanism. So on your screen you all can see the classification of passive transport mechanism. The passive transport mechanism does not utilize energy in the form of ATP or in any other form to get transported. There are 4 different types of passive transport mechanism, that is passive diffusion, port transport, ion pair transport, and carrier mediated or facilitated diffusion. So we will start with passive diffusion. Passive diffusion is the most important transport mechanism of drug because 90% of drug which are given orally transported through this particular process. Passive diffusion is also known as non-ionic diffusion because only unionized form of the drug can be transported. Ionized form of the drug cannot be diffused via passive diffusion. So, if energy is not required, what is the driving force? The driving force is concentration difference on the either side of the membrane, concentration gradient on the either side of the membrane, which means that the drug will transport it from the region of higher concentration to the region of lower concentration. When drug is ingested, GIT have higher concentration and plasma will have lower concentration. The drug will move from GIT to plasma. Right. So this passive diffusion is well expressed by Fick's first law of diffusion. Fick's first law of diffusion states that the drug will diffuse from the region of higher concentration to the region of lower concentration until the equilibrium is achieved. The mathematical expression you can see on your screen. It states that dC upon dt, it is the rate of drug diffusion, is directly proportional to the surface area A, partition coefficient K m by W, and C GIT minus C. This C GIT minus C is the difference in concentration gradient in GIT and C is plasma. Right, and it is inversely proportional to the thickness of the membrane. So, if surface area is more, the absorption or passive diffusion will be more, right? So, in case of small intestine, due to presence of microvilli, drug will transport more from small intestine than from stomach. The partition coefficient is a function of lipophilicity. So, if the drug is lipophilic in nature, you know like dissolve like, the membrane is also made up of phospholipids. It is also lipophilic, so drug will diffuse more. Now, the most important is CGIT minus C, that is the difference in concentration in GIT and that of plasma. The difference, ये जो difference है, जितना ज़्यादा होगा हमारी drug उतनी ज़्यादा transport होगी. राइट, GIT का कंसंट्रेशन जितना हाई होगा ड्रग का ट्रांसपोर्टेशन उतना ही हाई होगा. सो इट मीन्स दैट ये डिफरेंस होना बहुत ज़रूरी है. राइट, सो व्हेन द ड्रग इज इंजेस्टेड मीन्स जब आप ओरली ड्रग को लोगे, द कंसंट्रेशन दैट इज CGIT, concentration in GIT is much more than the concentration of drug in plasma, which is represented by C. It means that the drug will diffuse passively. Right, कब तक diffuse करेगी? When the equilibrium is achieved. So, if the equilibrium will achieved, what will happen? The process of diffusion will stop because the concentration on both the side of the membrane will be same. It means that larger amount of the drug will remain unabsorbed in the GIT only. So, do you think this is possible? Do you think it is worth it to waste a large amount of the drug? So let me tell you what exactly happens in passive diffusion. What exactly happened is that equilibrium will never attain. Why? Because when the drug is ingested, the concentration of GIT will be high and the drug will transport passively or diffuse passively from the higher concentration, that is GIT, to the lower concentration, that is plasma, as you all can see on your screen. Right, drug will travel from higher concentration to lower concentration, that is GIT, to plasma. Once the drug will go to systemic circulation, the blood is in circulation. It means the blood is flowing. It will swap out the drug from the site of absorption. right? And the concentration will again become zero in plasma, right? At the site of absorption, the concentration will become zero. So the concentration will always be high in GIT and the concentration will always be low in plasma at the site of absorption, and the process will never attain equilibrium. It is similar like the sink you all are having at home. The sink, when you will open the tap, the water will keep on flowing. Equilibrium will never get same. So whenever we increase the concentration, diffusion will increase, because it will diffuse and will continue with the process of higher to lower concentration, and this whole condition is known as sink condition. In sink condition, equilibrium will never ever attain, because the blood is flowing. The blood will swap out the drug which comes to systemic circulation, right? So the concentration in the plasma will be zero and the concentration in gastrointestinal will be high. So this was all about passive diffusion. Let's move forward to the second transport mechanism in passive transportation is pore transport. In pore transport, if the drug is aqueous soluble, it has chain-like or linear structure and has a lower molecular weight, which is less than 100 Dalton, it can pass through the water-filled channel present in the cell membrane. There are a number of water-filled channels which are present in the cell membrane for the efflux of water or aqueous-soluble nutrients like glucose. So such kind of drugs also can transport it through these pores, right? So this was pore transport mechanism, as you all can see in the diagram also. The next one is ion pair transport mechanism. So in the passive diffusion, what I have told you in the first introduction, that passive diffusion only take place if the drug is unionized in nature. But we have seen that if the drug is ionized in nature, it is also diffusing passively. Why? Because the ionic drug make complex with the endogenous ions present in the gastrointestinal tract. And this complex is neutral in nature, that is unionized in nature, and it will diffuse passively, right? So this is ion pair transport, where the drug ion, it makes a complex with the oppositely charged endogenous ion. Endogenous ions वो ions जो हमारे बॉडी में प्रेजेंट है, हमारे GI फ्लूइड में प्रेजेंट है. ड्रग उनके साथ एक कॉम्प्लेक्स बना लेगी और ये कॉम्प्लेक्स ट्रांसपोर्ट करेगा कैसे? पैसिवली. द नेक्स्ट ट्रांसपोर्ट मैकेनिज्म in passive transportation is carrier mediated transport mechanism. In carrier mediated transport mechanism, as the word implies, we utilize a carrier, right? So the drug will make a complex with a carrier. It will get attached to a carrier, and this carrier will carry the drug to plasma and release the drug in the plasma, and the carrier is free to take another molecule of the drug, right? So this is carrier mediated transport system. In case of passive diffusion, if you will increase the concentration, passive diffusion will increase. You can see in the graph as well. But in case of carrier mediated, if you will increase the drug concentration, carrier mediated transport system will not increase. Why? Because carriers are limited. Saturation will attain, steady state will attain. The only transportation is possible with the number of carriers present at the absorption site. Right, so it is dependent, it is number dependent transport mechanism, carrier dependent transport mechanism, right? So this is carrier mediated transport mechanism. In carrier mediated transport mechanism, one more term is facilitated diffusion. Now facilitated diffusion, unlike passive diffusion, it is a downhill process, which means from lower concentration to higher concentration, or the drug can move with a lower concentration only. How does this happen? This happens with the help of intrinsic factor. The intrinsic factor will form a link between drug and carrier, will help the drug to make complex with the carrier in case the drug does not have a structural resemblance with the carrier, and that complex will release the drug to the plasma, right? So this facility of intrinsic factors is termed as facilitated diffusion. So this was all about passive transport mechanism. Coming to active transport mechanism. In active transport mechanism, it is opposite to passive transport mechanism. It utilizes energy in the form of ATP during the drug transportation. Active transport mechanisms are of two types. First one is primary. In case of primary mechanism, the drug will transport with the utilization of ATP directly on itself, right? In case of secondary transport mechanism, the drug will transport with the utilization of energy, but not directly on itself, but indirectly, right? As you all can see in diagram, in primary transport mechanism, drug will utilize the ATP directly, but in secondary transport mechanism, the drug will not utilize the energy directly. It means secondary transport mechanism, endogenous ions will utilize the energy, and with the help of opening of channel, drug will also move to plasma. If the movement of endogenous ion and the drug is in similar direction, it is symport. If it is in opposite direction, it is antiport, right? Now coming to paracellular transport mechanism. In paracellular transport mechanism, the transportation process takes place between the cell membrane through the tight junction, right? So if the drug is permeating through the tight junction, for example glycosides or insulin, right, so it will transport it through the tight junctions between the cell membrane. And if it is persorption, it means shedding of the membrane. Sometimes the membrane will shed its cells, due to which the drug will transport through the shedding, right? The last one is vesicular. One more thing, paracellular is not of much importance in case of oral transportation. Now vesicular transport mechanism. So if the drug size is of macromolecule, is of larger size, vesicular transport mechanism come into light. In vesicular transport mechanism, the plasma membrane, or we can say the phospholipid membrane, cell membrane, will capture or engulf the drug in a vesicle formed by the plasma membrane. This engulfment is known as endocytosis. The drug is transported through the vesicle, and the vesicle will open the drug and release the drug in the blood circulation, right? So this process is known as endocytosis, and these are of two types based on the state of the drug. If the drug is liquid in state, the endocytosis is known as pinocytosis, and if the drug is solid in state, it is known as phagocytosis, right? So this was all about revision of mechanism of drug transport, and I hope you all can get the recap of the mechanism of drug transport through various processes, right? Transcellular, which is active and passive, paracellular, which is of least important, and vesicular, which is pinocytosis and phagocytosis, right? So I hope you enjoyed the session. In case you have any kind of queries, any kind of questions, any kind of confusions, you all can post your questions directly on the WhatsApp group of the subject WhatsApp group, or you can message me personally. So thank you for your time and stay safe.